jobs. First things first, though, we would like to acknowledge the traditional owners on the land in which I'm speaking to you from, the Wurundjeri people of the Kulin Nation, and we'd like to pay our respects to elders past and present. I'd like to extend this acknowledgement to all of the countries on this continent that we carry our work on. First Nations people are our first ecologists and biologists with tens of thousands of years worth of biodiversity knowledge. Through their stories, they have catalogued biodiversity and ecological data that we can only hope to complement and empower here at EnviroDNA. Okay, so who are we here at EnviroDNA? We're a small Melbourne-based company that have been founded by academics that are passionate about ecology, conservation, and knowledge. We're a small team of just about 25 of us, but we have a big client base of over 200. And these clients are based all around Australia, but also abroad. Our team is mainly comprised of field, data, and molecular scientists. We also have help from Jackie, who does our marketing and communications, who you already met. And we also still dip our toes in publishing with over 250 scientific publications under our belt. So we work out of these purpose-built labs in East Brunswick, Melbourne. To those who are local, you'd know that suits me just fine because the coffee around here is phenomenal. We've purpose-built 4 separate lab areas that are designed to minimize contamination and have a good directional workflow. And we also have a really high level of automation. So we have 1 liquid handling and 2 DNA extracting robots. And the company promises that they're not going to take my job. But in all seriousness, this level of automation allows us to process up to 1,000 samples a week, which can even be expanded, and it helps us do it as cleanly as possible. So here's a little look into our labs and our daily workflow. So to start off the session, we want to start with a poll, trying to just get a read of the room with where people are at and their understanding of eDNA. Hopefully Jackie is hitting go on that poll right now.
Let me just enable that one. Okay, our first poll. So hopefully everyone can see this. The question is, how much experience do you have working with eDNA? So we've got new to eDNA, I've used it before but still learning, experienced user, and then the last one is, I probably know more than Haylo. We just got a few answers coming in still. It looks like the majority of the room has used it before, but they're still learning. Okay.
Anyone know more than me?
We do have a few people that know more than you. 3 have to that.
They can buy me a coffee. It's fine.
I'll share the results so everyone can take a look. Okay. All right. Thanks everyone for submitting. It's always good to know where everyone's understanding is at before we go on.
Right. So for those who are completely new and fresh to eDNA, eDNA is shorthand for environmental DNA, which basically means that it's DNA that's found all over our environment instead of being sampled directly from a source. So an example of being sampled directly from a source would be straight out of a tissue sample. So how does eDNA come to be, and why is there DNA floating around? For those pet parents, you'd know how much cats and dogs shed hair, and it's kind of like that. Like, we're kind of just shedding it into the environment. We're shedding cells, we're losing them all the time. And all organisms kind of do this. They shed DNA. Some shed more, some shed less. Turtles are kind of notorious for shedding a really small amount of eDNA. And yeah, all this DNA shedding can kind of form a kind of DNA soup, especially in waterways, which is the most traditional eDNA sampling method. So traditionally, eDNA is sampled from aquatic environments. This is mainly because A, it's easier to sample water and feed water through a filter, but also B, movement of water kind of homogenizes that DNA soup and means that a smallish sample of only a couple liters is actually a pretty good capture of what's in that waterway. eDNA also degrades in the environment, usually within days in water. So that provides really good temporal data. So what you found in an eDNA sampling is most likely something that was there within the last couple of days. To expand on that, eDNA sampling in water is pretty simple. So in that middle picture there, you can see one of our field scientists, Josh. He's pulling up water from a waterway using a syringe, and then he just feeds it through a little filter that connects to that syringe. So sampling can be super simple. We also have a bit more technical automated setup. So Will, that third picture, he's got a pump that's pumping water through a sample, but yeah, it makes it pretty accessible to sample eDNA. However, just like we did, several millions of years ago, eDNA is kind of growing legs and moving into the terrestrial zone. And we're starting to learn about terrestrial sampling, where we're even just trying to suck DNA out of the air nowadays, which is really cool. But I think sampling from water is more relevant to this talk. So I'd probably, just talk more to that. Okay, so we've sucked up our DNA soup out of the water. What do we do with this soup next? What's next in the kitchen? So we have our usual pipelines in this lab here, where we extract DNA from the filters that you fed the water through, or even RNA if applicable. And then, we kind of make alphabet soup then. We've got all our DNA in there. From here, there are kind of two main analyses performed by us. The first would be target species assays, and the second is biodiversity assays. So the first is what you're seeing now on the screen, a little schematic of it. So that's our target species assays, or otherwise known as qPCR, or quantitative polymerase chain reaction. You probably heard that a lot to do with COVID, and it's a similar method that we use. So these target species assays are designed specifically for one species. So we find a region in whatever species we're trying to look for's genome that is variable from other animals, other species, and we design a probe around this highly specific and unique area in the genome. Basically, we throw it through a reaction with heaps of buffers, and I don't want to bore you with all the technical jargon, but at the end of it, if this probe picks up our target species, it will produce a fluorescence, and the machine that we have this reaction going in will quantify this fluorescence against known amounts of DNA. And we can get a really specific and sensitive readout from this kind of assay. And these kinds of assays also have a really quick turnaround time because they're pretty simple. They're just looking for presence or absence of one target species, which contrasts kind of pretty severely to our second kind, the biodiversity assays, or otherwise known as metabarcoding. And these are less specific by nature, and they're designed to produce a snapshot of a specific taxa or group. So for example, we can design one biodiversity assay that will amplify and pick up a gene that's present in all vertebrates, which is, obviously a huge scope of animals. We can get a little bit more resolution or drill down a little bit more and have just a fish or an amphibian assay, but it's to produce a really diverse snapshot of everything that's in the water. So how it works is basically we identify a gene that's conserved in a taxa, and we sequence all of the genes that are found in our eDNA sample. We get this little alphabet soup output here that are called reads, that are all the genes, and we feed it through a bioinformatic pipeline that compares all of our reads that we've gotten with a reference database. And basically we can be like, okay, we found sequence A in our pool, and the reference database has sequence A that's assigned to species A. So we can get a really good diverse read out of this. So it's kind of two different methods, one that's highly specific and highly sensitive, but one that's more broad and gives you a bigger breadth of data about all things that are in your sample. We can use these two assays, or these two kind of analyses, to measure life basically anywhere that life exists. So we can use it in biodiversity. We can use it in pathogen detection, like how qPCR was used for COVID. We can use kind of a similar thing. We can use it for monitoring restoration to see how restorative impacts are going. We can use it for environmental impact assessments, biosecurity, and also for the nature positive movement. So eDNA is really applicable across the board in all of these different applications. Now that I've gotten the base data down, I want to introduce you to SourceDNA. So SourceDNA is our brand new suite of offerings that are focused on source tracking for contamination in waterways. And it's using those two analyses, but in a pretty specific way. So it's designed to identify and track sources of wastewater contamination. The beauty of eDNA is that you can sample really easily and you have really good spatial temporal spread using that sampling. And it's also very modular, so we can design different target assays for different needs, and we can do different combinations of assays to really answer certain questions about what may be in a waterway that you are concerned about. We've also have this really great team of data scientists that have made this really accessible app that has all the data output, which Sarah will actually be walking through with you in a little bit. So what are some of the applications we envision for SourceDNA? So the first is to determine contaminated impact zones using kind of this app for the spatial temporal data kind of aspect of it, and finding where waterways near urban, industrial, and agricultural areas may be under some trouble. This can also inform management and investment decisions. Our second kind of idea for what SourceDNA could be used for is to assess biological risks. These are including human health and ecosystem function risks through identification of microbes of concern and their potential source. An example for this would be looking for gastrointestinal pathogens or bacteria in waterways to see if there's been potentially some spill. The third is to undertake impact assessments and to also quantify effects of management interventions in contaminated aquatic species, systems rather. So again, back to that spatial temporal kind of aspect of eDNA, we can use it to monitor temporally if our management and interventions are being effective. And we can also monitor pre and post discharge to waterways and assess potential contamination there as well using SourceDNA. Now I just want to walk you through kind of how SourceDNA works, how working through SourceDNA would look from a client perspective. And so with any project, we start with sampling. Rather sampling design. And we have a really great team of project managers who specialize in working through a client's experimental question and designing the optimal way to kind of answer that question. So they will sit down with clients and figure out, how many replicates would you need to have statistical power to answer your question? How many sites do you need? And maybe even how many times would you have to go back and sample those sites for temporal kind of resolution? And then the sampling is actually undertaken. You can undertake it yourselves. We make kits here that we send out, and they're really simple to use. So in one of those earlier slides, you saw Josh with a syringe in the waterway. That's one of the kits. So, and we make them, we send them out even to citizen scientists. So they're pretty easy to use, pretty easy to follow. Or we can even do the sampling for you because we have a field team who are equipped to do that. After the sampling is done, I get my hands on the samples in the lab. And we basically just do whatever analysis is decided upon between you and the project manager to answer the questions that you're asking. This is just a little, this table here is just a little example of what it could look like. So we can have a suite of target assays. So, we could have agricultural indicator species, or we could have human to see if there's human contamination. And then we can have indicators of waste contamination, like E. coli or Bacteroides that's human focused, that can tell us maybe if there's been some waste that's built in. So yeah, but that's also completely modular. So it might be that these aren't relevant to your question, and we'll have different assays designed for your question. We can also tie qPCR or target species assays in with metabarcoding and perform them both on the same sample. So we can have that highly sensitive target species assay, but we can also have the more broad metabarcoding or biodiversity data from the same site. Next, whatever analysis I perform in the lab produces heaps and heaps of data that is then handed over to the data science team, and they've put a lot of work into establishing pipelines that are able to handle the gigabits of data that we are spitting out at them and kind of make sense of it all. They have automated pipelines, but then they also have a set of eyes that QC, QA all the data and make sure that it's all above board and making sense. And then they actually feed it into our very fancy new SourceDNA app that provides both the reporting and the tracking outputs for our clients. The thing I love most about this app, and you're about to see it because Sarah's about to demo it, is that it shows everything very visually. And it makes it really accessible for anyone to really understand the story that the data is telling. So yeah, we have another poll here while I just hand over to Sarah to walk you through the demo.
Yeah, so while Sarah gets the demo ready, we do have another poll. So I've just launched this one. Hopefully everyone can see it. So if you're planning to apply SourceDNA, what application interests you most? We have human health, wastewater management, impact assessment, establishing baseline data, or other. If there's any other areas people are most interested in. Looks like the two key areas of interest so far are impact assessment and establishing baseline data. We do have a few responses for each of them, which is good. I'll just end it there so everyone can see the results. There are a few for human health and wastewater management as well, though. That's good. Okay, great. I'll stop sharing that. Over to you, Sarah.
No worries. Hopefully everyone can see live demo here. Thank you, Haylo, for the good sort of setting the scene of what the app is about. As Haylo mentioned, we want this app to be able to tell the story. So eDNA, particularly when you start using multiple assays, can become quite complex data sets. We were continually hearing from clients that we want something that is user-friendly, able to make decisions. Often it's project managers that don't have eDNA backgrounds or more in sort of on-ground works and larger program planning. And so how can we develop something that also fits a part around community engagement? These tools are being used heavily in community engagement as well. So hence why SourceDNA was developed. I'm going to just step you through it pretty high level here, but obviously reach out to me if you want a more detailed offline sort of step through of it. As you can see here, we map up your areas of interest. We can put on backgrounds such as waterways, anything that's really relevant. We can filter by catchment if we want. The black outlines are obviously the catchment. If you have particular programs or site codes, and we can certainly filter it by that. For this one, this part, or like I guess example demo, we've got metabarcoding and qPCR. This one is very much from the lens of looking at I guess sewerage or fecal contamination. So this will give you a good sense of how it can start telling you a story. If we look at metabarcoding, a general bacteria was run on this particular example. We're able to flick on indicator species. So what we looked at is in the bacteria results, the DNA results, we looked at what would be indicator species. And we can switch those on. You can see down here that it's species richness. So you're looking for those that have got, high numbers of species. The purple detects where there's not many. So we see sort of this area down here, and up here are reasonably light on. We know that we've got some hotspots around here by just looking at metabarcoding indicator species for bacteria in terms of fecal contamination. We can switch it to qPCR. Now, this is where the story starts to come into it. If we just switch on qPCR, this is for all the qPCR that's ran. We ran Bacteroides looking at human-specific Bacteroides, E. coli, again, more human-specific E. coli. We then ran human, poultry, dog, sheep, cow, and pig to understand whether that contamination or those high loads of bacteria, Bacteroides, E. coli, may be associated with other species rather than humans. And so we start to unfold this story. What we can do here, you can start to see, so you've got high loads of Bacteroides all through here. You start to make a sense of where some of the high areas are. What is quite helpful is just flicking it through. So for Bacteroides, again, your log down here is looking at yellow is high. We start to tease out some of those high problem areas. So again, this particular area is obviously of high focus. We have a look at what that looks like against E. coli. Again, there's a pretty good correlation. And if we flick back to the bacteria, there would be a strong correlation again around this particular area. These high loads, say of E. coli or Bacteroides, don't necessarily mean it's human. So we start to try and understand, okay, is there sheep in any of we know up here as we go up the catchment in this particular scenario, there's probably more rural areas. We're starting to see that filter down. So the waterway is starting to tell a bit of a story down through here. It's likely that, you've got agriculture up here and you're getting some level of impact. No influence from sheep down here. We look at poultry. Again, lots of people love to have their chicken coops, particularly in kind of urban and semi-urban areas. We start to see that poultry is playing a bit of an indication too through here. What about pigs? Nothing. So we know we rule that out as any particular source of interest. We have a look at human. We know it's aligning similarly to if we flicked on E. coli. We've got this hotspot down here. So we're starting to understand that a little bit more. We have a look at dogs. Again, just people out, not cleaning up after themselves, or it's just, wash off from the streets. What does that look like as well? We've got a really nice hotspot up here. Something's going on. You can kind of drill down. You could zoom in, have a look. Is there, does it make sense in terms of like, is there a dog park, whatever it is it high urbanized area? And we obviously cattle as well. Again, no cattle in this. So from an agricultural point of view, we've got sheep playing a role in the system, and we've got some poultry influences, and we're starting to see that relationship in humans. So if we were going to use this as and we'll talk about the different ways that you can use it. But for human, with say high Bacteroides and high E. coli, I'm flicking a lot here, you know this is your target area. You're starting to really tease out some of your problem areas that if you were going to target and use as priorities, you might go into those areas. So what we're wanting to do is tell a story down a catchment. We use qPCR because we can understand, I guess, each sample in relation to each other. And I guess the loads of DNA that would be in each one. And so are we seeing it? Yeah, so you've got a subtle influence all through here, but it really spikes around here. And so this is where project managers and sort of, waterway managers can start understanding these trends. We have a simulated data here. What this app can also be used for is to show change over time. So if you were to implement a strategy, say around this area here, what would it look like as you took sort of ongoing monitoring? You've got a really nice baseline set here. What you hope is that it will be improvement over time. We sort of have this play where it changes over time. You can see the colors subtly changing with the different data points. And this one's kind of showing that, some good management has meant that this has gone back to sort of relatively normal background kind of, or what you would expect. And so, I guess that gives you a sense. We have a lot of data that sits behind us. You're able to download it. We also obviously provide a report and the data, but that gives you a sense for what it can show you. I'm going to step you through a couple of case studies around how the app can work. As Haylo said, it's very modular. You'll see that our 3 case studies are so different, and also the SourceDNA app itself can be adapted to. It was really important for our initial clients that they had that simulated data or that change through time. But for other clients, there may be a different need or a different way to illustrate the data. And so it's flexible. We have a wonderful data science team that works through these apps. And so, certainly as a project manager, we will work through that, what the app actually needs to look like and whether any changes need to be made. So I'll just stop sharing and Haylo's going to reshare the presentation. We've got 3 case studies doing a program with Yarra Valley Water. I think probably quite a few of you on the call will have worked with Yarra Valley Water before. They have a community sewage program. As part of that, they are upgrading, areas that are not connected to sewage infrastructure to new sewage infrastructure. So it could be from septic into sewage infrastructure. One of the things that they are hoping to use, or ultimately, planning to use the data for, is to inform the works program. Where are we having those hotspots? Where do we need to, whilst they're being done as a package, where do we need to focus that investment really early on to get kind of probably the bulk of the impact sorted, and then over the next 5 to 10 years roll the rest of the package out. So it's being very much used in that project planning phase. Also understanding that the regulator needs to see that there is improvement over time, and so how do we show that? And hence why that change over time becomes really important, and collecting baseline is so important. Melbourne Water has a really a powerful, baseline data set for the Melbourne region, and Yarra Valley Water has been accessing that for some critical baseline data for a couple of years before they even started this program. So it's really exciting to see how eDNA as like a biobank can be used as well. In this one, we're looking at very similar suite to what we presented. So looking for just a general bacterial metabarcoding, and looking for indicator species and the relationships between host species to try and just tease out where those hotspots are. One thing I didn't actually show you on the app is these heat maps. So we saw on the map that there was a couple of dots. So this yellow dot suggests it's high. You can sort of heat map it out, and it's rather than just trying to piece the story together with the dots, you're able to see those hotspots. And that heat mapping can be, I guess, configured to the question that you're asking. What relationships mean that would be high, and then we can configure the heat map on that one. The next one that we're working on is in the human health section. So again, really different application. Whilst sewerage obviously is does have human health implications, this one is looking at a really different suite of human health indicators. It's around gastrointestinal pathogens in remote communities of PNG. So looking at viruses, bacteria, parasites. We're using combination of, this is currently in progress. The sampling has just been completed, and we are using a suite of qPCR as well as metabarcoding to understand that the drinking water is accessed by nearby waterways through communal faucets. The sampling was then taken at the faucets to understand, what gastrointestinal pathogens are present in the eDNA samples. Understanding, and you'll see here, it is north of Port Moresby by quite a way. So if for those familiar with PNG, it's around Lae. The team mobilised from Lae, but still very remote from there. Someone hiked over a couple of weeks to get all these samples, and it was a pretty mammoth effort by Zoo Miami, who we're working with. You can see some photos here. It took a lot of planning, and it was executed really well. They've had to take some portable refrigerators just because we were dealing with viruses and RNA. We wouldn't normally have to do that with eDNA, but there was an additional element there. So it was a great opportunity to get the communities involved. Again, one of the key reasons why SourceDNA was chosen for this was the ability to present the data in a story in a meaningful way. The local, not local doctors, but doctors that will come in will access the data as well to just try and understand spikes in different communities, and also be able to present it to community members in a visual way as well, that without having to delve too much into the technical details. So for this one, it's part of the One Health program off the back of COVID, and it's funded through that, and just trying to understand how risk and I guess trends change with pathogens throughout a really remote catchment. The next one, and this is the last one I'll go through, we're working with another water authority in Australia, and we're using it to look at invasive species. So some of you will already be collecting a wide range of data, through water quality kind of testing, whether it's, chemicals, metals, E. coli, a lot of people do. So in this instance, it is in drinking water catchments. We're consistently seeing some high E. coli readings. We are not going to run the E. coli on this project. We're going to utilise the existing data they have and bring it into the app. We're going to run metabarcoding for vertebrates as well as that SourceDNA suite of potential host species. Or source species. So the reason we're going to broader vertebrate is to pick up anything else that is understand more broadly, I guess, what's hanging out in those catchments, but also to pick up things like deer, any other invasive species that we just mightn't have quite, that is not pulled into that suite as yet. Might become part of an ongoing monitoring, but just to give a sense of, is there something else at play that's causing these high E. coli readings in some areas? So a nice way to bring in existing data, and meshing it in with eDNA in a visual way as well. So that, yeah, I guess that is, I'll just get you to go back, Haylo. That is, I guess, all the case studies. As you work through your question, we certainly can work through whether SourceDNA would be a great output to start visualizing and monitoring over time. So I guess there's a bit of a summary for this webinar. We know that currently a lot of that human health and ecosystem function contamination monitoring is done by chemical properties and bacterial culture-based approaches. But eDNA can play a role in this ongoing monitoring. You can use it to identify and track biological risks and impacts from contaminants. We see, I guess, SourceDNA is a first step, a really small step, but a really important step, and it's very much focused on that biological risk. In the future, we really hope that toxicity studies are going to start bringing in biological data a lot more, starting to really understand what does these ecosystem services, limits limits for each species. What does that actually mean in reality? And we're starting to get some really great baseline data that we can start teasing this information out. But understanding biological response to contamination, I think that's where, we hope that eDNA will move in the future and be a really nice complementary data set to that really traditional chemical sort of culture-based approaches that are currently being used. I think that is it